Hello, everyone. This is Kiwoo Kim from the College of Dentistry of Yonsei University. We have been studying whole body metabolism is controlled about how the central part consume through the autonomic nervous system, not the peripheral part. We have found interesting materials, and we decided to IP transfer some of the materials that has a potential to be phenolic derivatives and peptide drugs. What we are studying for are eating disorders. Eating disorders can be categorized in three categories. If you look at the red letters on the right side of the top, anorexia nervosa, bulimia nervosa, and binge eating are the symptoms that has been pointed out as the biggest problems of eating disorders. We not only study eating disorders, and if you look at the screen, you can see obesity population that include people with obesity and diabetes in 2004 and 2007. Also that the rate of metabolic diseases will increase geometrically if you look at the prediction chart in 2014 and 2035. This is why the increase of metabolic diseases and eating disorders have been the greatest concern in our laboratory. We had been studying electric generation of low energy in the past. There are decent metabolism modifiers in the current market. Anti-diabetics are focused on lowering blood glucose, and obesity modifiers focus on controlling body weight maintenance. Current most used drugs are effective, but there are many side effects, as you can see on the purple panel of side effect. The side effects even include increase of suicide rate. On one instance, Belvid has been eliminated from the market this year because it causes cancers. We have been studying safe drug development with few side effects as well. We are studying a lot about mechanisms related to obesity, especially from all the metabolic diseases. And let's look at MOA of FDA-approved drugs or mechanisms. On the left panel, you can see drugs listed from phentermine to liraglutide. On the MOA panel in the middle, all the drugs except for orlistat, like sympathomimetic, lorcaserin to C agonist, and phentermine topiramate modulate the central system, such as GABA modulator. This suggests that obesity is not just an obesity, but it can be categorized as a symptom of mental disease related to the central system. And I'd like to say that it's hard to develop drugs for obesity without understanding brain nervous system. We simplified the subject that we are currently studying in our laboratory, and on the right side of the bottom, you can see it's written HCR. ACR means hypothalamus. Hypothalamus is a center that controls physiological things, especially appetite, libido, and circadian rhythm, out of central nervous system. Among them, the ARC written POMC neuron and AGRP neuron are important parts of central neural system and energy homeostasis. So those two neurons are known to play significant roles. The basic mechanism is that when AGRP neuron gets active, food intake increases and energy expenditure decreases, and this results in getting weight on the whole body. For POMC neuron, when it's activated, it increases energy expenditure, and this leads to weight loss. This is the most known study so far. The reason why I'm telling you these is because my materials that IP transferred affect central nervous system. So, I will address these briefly. When we were designing drugs, we used this as a reference. It says drugs have been developed focused on low molecular weight chemicals with high effectiveness from 1960s to the early 2000s or 1990s. But recently, with metabolic diseases, the drugs had been just focused on effectiveness, but had too much side effects, and people have been dying from it. So protein or peptide drugs have been developed that value effectiveness and safety. You can see that synthetic peptide has more pipeline than low molecular weight chemicals on the bottom panel. So, according to this trend, we studied central nervous system when we did target screening, and we were interested in phenol derivative with high safety and endogenous peptides produced from our body. We found materials that control central mediate body weight, blood glucose, and energy expenditure after the screening. Today, I will address phenol derivative 2, and peptide. First of all, let's look at PD1. This is also publicated, so you can find out by looking for the paper. To tell you the visions, looking at the two panels on the right of the 5 figures, PD1 showed that AGRP and NPY neuron perform dose-dependent suppression in AGRP neuron. This plays a role in reducing food intake, and if you look at the first graph on the left, black is PD1. When it was given high pass, lipid contents give high Western diet, and when PD1 was injected, it only gave Western diet, and we could see that around 10% of body suppression was happening. We also conducted glucose tolerance test, and you can see that glucose tolerance has improved in the panel in the middle. PD1 can be derivative that targets NPY neuron. What we found secondly was more interesting, and it was that has a function of AMPK suppression in hypothalamic. Let's look at the graph on the right. AMPK is used as a treatment for diabetes. It lowers energy cellular level in peripheral and increases insulin sensitive. When AMPK gets active in the brain, it changes to hunger signaling. This is why AMPK works as a bad way, as it makes people eat more and gain weight. But the derivative we found can suppress AMPK activity, and it increases glucose tolerance or insulin sensitive and suppresses food intake. We named this PD2 and conducted IP submission. Thirdly, it's endogenous peptide. It's at the last stage to submit IP. To start with screening, it's endogenous peptide and can act as a neuropeptide that's produced from our body. So screening peptide when processing, if you look at the pictures, right under the various changes of them, and that there is no change of the chest wall, is at the bottom of the picture. So, when we treated brain to a mouse model, food intake was suppressed and body weight decreased. The red is peptide that we target. Before finding its mode of action, we conducted a test if it increases glucose tolerance and insulin sensitivity while controlling food intake and body weight, and we found that it does increase insulin sensitivity. We were so interested in checking its mode of action, as it's an endogenous peptide, so we treated it and measured activity of neuron. You can see the result in the pie chart on the bottom. When POMC neuron gets active, it suppresses food intake. We've found that neuronal activity was activated around 41% in POMC, and we've dug into this deeper thinking endogenous peptide could be some sort of target of POMC neuron. We prepared animal models that can label POMC neuron, and if you look at the date on the left bottom, when treating JP, resting membrane potential increased slightly, and when hitting JP, spike getting denser. This means when peptide is treated, neuronal activity and caloric ring increases. In other words, endogenous peptide can activate POMC neuron. The next thing we did was move screening M4D1, which is a dread system that can suppress neuronal activity, to find out if POMC neuron is required. When POMC neuron was planted in this and give a compound called CNO, it can suppress neuron mechanism. In this situation, I gave JP and measured food intake, and we could see that the phenomenon of food intake suppression was gone. We could also see that the beneficial effect of increasing glucose tolerance disappeared. What we think is that POMC neuron is necessary for endogenous peptide to show these effects, and we are conducting characterization through LCMS and pull-down assay to figure out its binding partner and receptor. When the characterization is done, we can explain you through which receptor endogenous peptide activates POMC neuron and control food intake, body weight, and energy homeostasis. The major material of our study is JP peptide. We think this peptide drug can result in a good metabolic consequence, as it activates POMC neuron and has an effect of anti-obesity, anti-diabetes, and blood sugar control. Thank you for your attention.